Good afternoon, everyone. I'm Colin O'Connor, Custom Content Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Single Cell Genomics at Memorial Sloan Kettering Single Cell Analytics Innovation Laboratory, and it's sponsored by Levitas Bio. Our speaker is Ronan Chaligne, Director of the Single Cell Research Initiative and Assistant Lab Member at the Sloan Kettering Institute. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Ronan.
Hi, good morning, everyone. Thank you for joining. It's a bit strange to present alone in my office. I would really love to be in front of an audience with everybody in front of me, but I'll do my best to keep you entertained during this next hour. So I've been director at SCREE for like about a year and a half now. Just to give you a tiny bit of my background, I am French, as my accent is probably telling you now. I did my PhD and the first postdoc in France, where I study cancer and epigenetic. And then I moved to New York, where I am now, where I did the second postdoc in Dan Landau laboratory, where I developed 2 single-cell methods and study blood cancer and develop single-cell methods. And after that, I moved here, where I am now director of single-cell research initiative. So my background is really wet lab. And a lot of the things I'm going to present today are focused on wet lab, on the experience the team I'm managing has in trying to handle clinical sample or like model organism to perform single cell genomics. And I'll try to give you a nice overview of what we do. So basically, short title for today would be how to single cell in a wet lab. And the first things I want to introduce in this presentation is the team that's doing the job every day, because I am director of SCREE, but the one that are really important in generating good data and analyzing those data is the team that you have in front of you. So for now, we have a team of about 10 people, and one of our specificity here is that we are a mix of a wet lab and a dry lab team. So half of the people working at SCREE are handling sample, working on optimizing, benchmarking, sample preparation, or new single-cell genomics method. And the other part of the team is a CombBio team, which is like processing the data, doing analysis, and also benchmarking and optimizing method, but in the dry lab. So we're kind of proud of being this kind of mix of dry and wet lab, and we really try to innovate in both worlds and try to do our best to get like a nice biological question answer with single cell genomics. So our main mission, and this is thanks to Memorial Sloan Kettering, where we are located, our main mission is to basically embrace new single-cell technology to answer new biological questions. So we really try to push the boundary and do single-cell genomics method to discover new signal or new biological information within the sample that we are handling through collaboration. Just to give you a quick overview of how much single-cell genomics is rising, and probably if you are here in the audience, you know that because you're interested in single-cell, is that you'll see in a minute. It's a pretty old, I would say, technology, or something that researchers have been chasing for some time to do single-cell transcriptomics analysis. But it has really been like rising and developed over the past 5 or 6 years with some like commercial platform that are really make it efficient and more high throughput. But to speak about Cell, so I mentioned SCREE before. We are changing names, so now we are becoming Cell, which is Single Cell Analytics Innovation Lab, but we are the same entity. We are just getting rebranded. So at Cell, we've been existing since 2017. And these are like just few number of how many samples we have been handling and how many single cell we have been sequenced. But most importantly, that's the number of samples we're handling every year that keep rising. And this really show at our really local level how much the community, how much the research community is interested to do more and more single cell. And what we see every day that people that have no experience, no previous history of doing single cell work are now starting to do single cell genomics analysis to really get into more detail in their research project. So one of the motto we have, we are not really a core facility because our motto is fail for first attempt in learning. Like we, as I say, really try to innovate, and innovation means failing. We try to praise ourselves with not being afraid of failing. We actually feel that failing can be good because we learn something out of it. And that's what I'm going to try to show you on everything we have been working on over the past few years. But to start with, I just want to really briefly touch base on the history of single cell and show you when the first paper trying to study single cell has been published. And it's pretty old actually because it's from 1992, where they look in single cell doing 1,000 blot assay, where basically they start to extract RNA from single cell, from neurons, and they do amplification through PCR, and after they try to interrogate which transcripts are present by doing 1,000 blot. So you can clearly see that it's pretty low throughput and not really quantitative at the end of the day, but it was really the first attempt of trying to assess what genes are expressed at the single cell level. Few years later, I would say almost 2 decades after that, the real kind of higher throughput single cell method has been published by the lab of Azim Surani, where they look at the mouse blastomere, which is kind of a big, which is kind of early development of mouse, and cells are all full of RNA. So it's kind of a model system where you can get a lot of RNA content, which make it maybe easier to assess single cell transcriptome in this kind of model, which was kind of really smart in terms of technology speaking to look at those. And they, for the first time, assess full transcriptome of single mouse blastomere by doing at the end of the protocol next generation sequencing and look at all the transcript they generate. And you can see that they found that about 1,500 genes that were abnormally upregulated compared to the wild type control, because they look at different knockout oocytes. And this was really the first example of single cell full transcriptome method. Of course, now there is more and more commercial platform becoming available. The first one, I think, to my knowledge, that's become available about 10 years ago was a Fluidigm C1, which tried to capture single cell and making more high throughput. But now we know that the one leading the market is a 10x Genomics product, which has developed a variety of single cell genomic approach from single cell RNA-seq, single cell attack, which has a chromatin landscape, or even combine where you can do attack plus RNA from the same cell. There is other commercial platform on the market. They all have their specificity. For example, the Illumina Biorad is more focused on doing single cell attack. The Rhapsody single cell analysis will be more targeted and maybe can be more sensitive than the Chromium 10x Genomic, but maybe will be less full transcriptome. You will only interrogate a couple or 100 of gene. And I think I cannot make the full list of everything that is here, but there is different platform, even though 10x is the one leading the market and done by most of the lab doing single cell genomics now, and by ourselves even. But I would say now single cell is becoming more and more used by many labs. The next, I would say, development in single cell will be to do single cell multiomic, which is basically now interrogating several layers of information from the exact same cell. There is like a couple of methods that have been developed, like in-house or I would say homemade method or like commercial method, but all the methods principally focus on mRNA and trying to interrogate other components from the cell. So we can see method where they look at lineage tracing. I can briefly touch base at the end of the presentation on this. Some method will assess surface protein or even now transcription factor or more like internal protein combined with mRNA. Some method can look at the SNV or indel happening on the genomic DNA. Some single cell methods are looking at demethylation, copy number alteration on the genome of the cell. Some method like single cell cut and tag, for example, can also look at histone modification, look at chromatin accessibility with, of course, the single cell attack. Some method like single cell Hi-C will look at the chromatin conformation and how two different parts of the genome are like interacting with each other. And of course, the next stage of all the single cell will be to add the spatial information to it with like the kind of development of many different platforms doing spatial transcriptomics. I'm not going to talk about it today because we are just starting and we don't have extensive experience with it. But definitely like a company like 10x Genomics or RNAscope or VisGen or NanoString are all developing their own platform to do spatial transcriptomics with different assays. And if you have questions about that, I'll be happy to try to answer them at the end. This is just like a small table to just really put emphasis on all the different methods that exist out there, trying to assess all these different components. And you can see that they all have their kind of limitation, but also their advantages in terms of throughput or what type of information you're going to get. Is it the full transcriptome? Is it the full genome? Is it just like a targeted part of the genome or the transcriptome? And also, like, do you want to combine information? Do you want mRNA plus some kind of lineage tracing? Do you want the mRNA plus some of spatial information? So I would say before deciding of which single cell methods is suitable for you, I would say the best you can do is really try to ask what are your biological questions, and I'm going to get back to that later. And based on this biological question, I think you can decide what's going to be the best single cell method that you want to move forward with. Because again, they all have their advantages and caveats. So today I'm going to mostly talk about 10x Genomics because it's our bread and butter. It's our basically daily activity. We mostly use their product. Several reasons is like most of the publication now using single cell genomics method are using 10x Genomic products. So I would say the single cell community knows those products really well. They know they are reliable and they know that the data you can get out of those methods are really good quality and can also generate a nice data set. So there are like three main, in the single cell genomics world, there are like three main kit. One is doing single cell gene expression. I'm not going to touch base on the different variety of single cell gene expression, but again, happy to answer a question if you have at the end. One rely on capturing the transcript from the 3' end, another one rely on capturing the transcript from the 5' end, and they both have their advantage and caveat. Second method is single cell attack, which is basically looking at the chromatin accessibility at single cell level. And they released a bit more than a year ago, a single cell called multiome, where from the same cell or the same nuclei, they are assessing RNA and attack at the same time. In all the cases, the big advantages of 10x is that it's relatively high throughput. You can go up to 10 or 20,000 cells. And in both cases, you can assess the full transcriptome or the full genome for chromatin accessibility, which is like allowing you to really kind of explore in an agnostic way what are the changes happening in your cell population. So there is several things based, again, on our experience. I'm not trying to make a list that's going to be completely kind of complete for everybody. This is just based on our experience on the things I've observed, like interacting with different collaborators at SKI and also a bit around the research center around MSK. The first things I want to touch base is a biological question. I think a lot of people are diving into single cell without a clear idea of why or how. And I would personally feel it can be then overwhelming, because when you get single cell data, you get a full transcriptome for every single cell you have access. And if you don't know what to look for or what to explore, it can be overwhelming. And just doing cell clustering won't be enough or won't be satisfying, I think, at the end of the day. So starting a project for single cell with a clear question is really a way to make it, I think, successful. And this is just, I'm just showing one of this example because I know it well, and I've from Mario Suva lab, and I've collaborated with him. Is because it's a paper published in 2016, and they do like SMART-6-2, a single cell RNA-seq, so it's not 10x, and it's a pretty low throughput method. But one of the really smart ways they have done here is that they, even though they don't get a lot of single cell at the end, because they only get in a range of 4,000, which might seem like a really low number for today's number, like 6 years later. But what they have done is like they study glioma, and they try to answer a question that was, is in glioma development, which is a brain tumor, is there like any transcriptomic profile or cellular state that would remind like a normal or a classical glial cell development? And they ask this question and then look at the transcriptome they generate from glioma, looking at different pieces from the transcriptome of each single cell, and they try to just answer this question. And even though they have like a relatively low number of cells, they manage to find that some of the gliomal cell will express a specific lineage marker that you can find also in normal astrocyte or oligodendrocyte development. So it was like a really nice way to, even though the data set is small, try to answer the question with the information you have. Second things I want to bring to your attention in things you want to consider when doing single cell genomics is keep in mind that before, except if you work on blood or hematological model system, where of course the cells are already in the single cell state, most of your model system will be tissue, and then you will have to, before doing a single cell genomic, have to dissociate and separate every single cell from each other, and there is like now a variety of ways to do it, but the classical one is to basically take your tissue and dissociate single cell by applying like enzymatic digestion on it for like an hour or like half an hour with a cocktail of enzymes that will be dependent of each tissue. So after that, you can generate nice single cell suspension that will allow you to do single cell genomics or single cell RNA-seq. But one of the caveats of this, and this have been, I feel, overlooked for some time, is that basically dissociating a tissue for like almost an hour with enzyme can be really stressful for the cell in the tissue and can create a lot of bias to the transcriptome of the cell you're going to recover, but also bias toward the cell population you recover, because some cell will be more sensitive to this kind of dissociation and might suffer from it or even die. So this is a nice paper I find, where basically they treat during dissociation their tissue with two inhibitors, one inhibitor of transcription, one inhibitor of translation, And what they show is that if you block the transcription activity during dissociation, you kind of prevent your tissue from, or your single cell from showing stress signal. So if you look on the panel C, you see that when they use enzymatic, so the upper right panel, when they use enzymatic without inhibitor, you have this kind of population, which they call ex vivo activated, which show like some stress signal, some like stress activity that's mostly due to tissue dissociation. But when they treat the stem tissue and now they do enzymatic with like inhibitor blocking the translation and the transcription, then all this population being stressed completely disappear, which means that because the cells are not able to activate their transcriptome or respond to this kind of stress, being dissociated, you don't see having this on your tissues that have been treated with inhibitor. So it doesn't mean you have to treat with inhibitor every time you go dissociation, because there is maybe some other caveat you are introducing doing that we are not sure yet. But I think this is a nice example to show how much doing enzymatic dissociation can stress your tissue. This is a second example that basically lead to the same conclusion, where they basically study muscle, and here they correlate dissociation, time of dissociation, with stress index. And if you compare the two panels, the G and the H, you can see that the longer you're going to dissociate your cell, the more stress they're going to be, which make complete sense. Like the more they stay at 37, the more you're going to treat them with enzyme to dissociate them in the tissue, the more chance you have to stress them and also change their transcriptome. So this is something you really have to keep in mind. And I know that because single cell method were not really sensitive in the past, maybe they were like not as good as today at capturing the transcript from the cell. People used to prefer working on cells and on nuclei. But you have to keep in mind working and dissociating tissue will have a big impact on the transcriptome of your cell. And just to finish on this topic, because I really feel this is important, and now on the work we do every day, I'm trying to push more and more to work on nuclei rather on cell. I just want to bring to you this paper, which is basically comparing doing single cell versus single nuclei. So I did not talk about nuclei, but the two ways you can look at the transcriptome of a single cell is either dissociating the tissue and get the full cell, but you can also take the tissue, snap freeze it in liquid nitrogen, and then dissociate mechanically the tissue and then just recover nuclei. Advantages of this method is that instead of having an enzymatic reaction allowing to separate every cell, when you snap freeze and you dissociate the tissue mechanically to get nuclei, your tissue is basically dissociated mechanically by just the action of smashing it once it's been frozen. And the nuclei are strong enough that when you smash it, you recover the nuclei of each single cell. And you don't have bias during the dissociation because it's mechanical and not due to enzymatic dissociation. So this paper is kind of trying to assess whether RNA-seq can be done on cell or on nuclei, and what are the bias induced in each of the methods. Because when you do on nuclei, of course, you are capturing the transcriptome only from the nucleus and not from the whole cell. So doing so, you are just losing all the transcripts that were present in the cytoplasm of those cells. So this paper from Aviv Regev group have compared different tissue. Here I am just going to show you this neuroblastoma example. And on the panel D, what you can see is like the proportion of each cell type that is recovered, either if you do all cell or nuclei full transcriptome assessment. And what you can see, for example, is when you do single cell RNA-seq, you tend to recover a lot of immune cell, which is the black bar graph in the cell. And when you do nuclei, you recover a lot less of those immune cells. And I think there is two reasons for that. The one is like when you do single cell dissociation with an enzymatic reaction, the immune cells tend to be the one that will survive better to this enzymatic dissociation. They are like strong and kind of not really easy to kill. So when you do single cell dissociation with an enzymatic reaction, the immune cell survive, while more fragile cells like epithelial cell or endothelial cell will tend to be more sensitive to this dissociation and will die way more quickly. So that's mostly why when you do cell, you probably recover more immune cells than the rest of the cell. And the second reason is that when you do single nuclei, you're gonna recover, as I said, just the RNA from the nucleus. And the RNA from the nucleus in quantity is way less than the whole cell. An immune cell compared to endothelial or fibroblast or other cells tend to be smaller cells with less RNA content. So when you do single nuclei, the immune cell nucleus will give you less RNA than an epithelial cell, for example. So by doing nuclei, you tend to also recover less transcripts from the immune cell, and you tend to capture less efficiently those cells doing nuclei. So you have to be aware of this bias if you want to go for single cell or single nuclei. I think the single nuclei tend to give you a better representation of what the cellular content is in your tissue, and also give you a better idea of what the real transcriptome is in your tissue, because you really snap freeze your tissue as long as you isolate it from the mouse or the patient or whatever your model organism is. But at the same time, you know that you're going to create some bias against the immune cell. Another thing they look in this paper is like the dissociation signature. And of course, the dissociation signature is way more present in single cell RNA-seq as in nuclei, as I was showing you before. But they also identify some of the stress signature in the nuclei. But they argue in that case that this stress signature is probably a real stress signature that was present within the tissue. And mostly in that case, it's probably reflecting immune activation, meaning that even when you do nuclei, you probably see some stress, but this stress is probably a real stress that was biologically meaningful because the cell was stressed within the tissue. And it's not a stress that is induced because of your methodology or your protocol, which I would argue is good, meaning that when you do nuclei, you might recover some stress signature that's real and not just artifactually created by your method. So I hope I convince you that nuclei is a bit better than doing cell when you have the possibility to do so. Something also to keep in mind when doing nuclei or single cell is the level of RNase activity you have in your tissue. A lot of method or like commercial, and that's true especially for spatial transcriptomic. I think today a lot of the company are going to show you brain data set, because the brain has a big advantages to have really defined cellular state, but also have like close to zero RNase activity. So you tend to recover really good transcriptional data set from brain, which is not the case when you do other organs or tissue. We have been extensively working with lung and pancreas here at Memorial Sloan Kettering, and we had to really use trick to prevent those tissue to get having the RNA in those tissue degraded by the RNase activity when we do either single cell or single nuclei preparation. So keep this in mind. Also, the tissue you are using might impact which protocol and which method you want to use to prevent RNA degradation. So something, because we are part of HTAN, which is a Human Tumor Atlas consortium that we had to handle like a lot of sample over the years, we have been kind of really refining the logistic. And I would say that every good project, beyond having a good and a proper biological question to answer, also start with a proper logistic. Knowing how, when you're gonna get the tissue and how you prepare it, how you're gonna do the single cell genomics method on it is really key to help you get good data at the end and something you can really use to analyze. So here I'm just trying to summarize the three ways we are preparing tissue and what are each the pros and the cons of those way of preparing the tissue. So one thing we have done, and this is kind of the way we have done for years, is what we call fresh suspension. So basically, and this is really something we are doing within the Human Tumor Atlas project, is that we are coordinating with the operation room and trying to get the clinical sample as soon as it gets rejected from the patient. After that, we'll have to do the dissociation and to do FAC sorting to enrich for cell population or select for viable cell only. And all of these things might take between 3 to 4 to 5 hours. And after that, we have to get ready to do the single cell, the beginning of the single cell protocol, which is the encapsulation of the single cell within the droplet. So all of these things take about 4 to 5 hours before we can reach a stopping point in the protocol. And it requires many different players to be really ready to go and handle the sample at a given time point. So this is a great way to do single cell if you have people ready to be handling the sample, and this allows you to get specific cell population enrichment. For example, if you just want to enrich for immune cell or like tumor cell or epithelial cell, you can, because you are gonna fact sort your single cell population. You also get single cell RNA-seq quality, meaning you get the whole transcriptome from the whole cell. And also this allows you, for example, to do site-seq, which is a way to interrogate the surface protein of your single cell. The cons of this is that it's really challenging logistically speaking, because you need to be, again, ready to handle the sample. And most of the time, clinical sample are dependent of when the surgery is happening, and surgery is frequently postponed or delayed. And then meaning that a lot of people can be ready and then it's not happening. So it's a lot of waste of time for everyone. And these are also the cons of having to dissociate enzymatically your tissue. Another way we have found to try to kind of circumvent this kind of caveat of making more easy on the logistic, it's to do what we call fresh frozen suspension. So we basically also get the tissue fresh, we do the dissociation, and then we can store it in liquid nitrogen for weeks before we start processing it. So this has a good advantage to not have to do the encapsulation on a given day, especially if the sample, for example, the surgery is happening late in the day. We can just take the sample, take the tumor, do the dissociation, and then freeze it in single cell suspension. On a given day later, we can just like sew it and then sort it and then do the encapsulation. So it's giving a bit more flexibility, still have the advantage of doing single cell and the caveat of it as well, but it gives a bit more flexibility in the logistic. But in that case, you just have to be aware that some cell type might not survive well being frozen. Like dendritic cell, for example, will not survive being frozen in liquid nitrogen. So again, it's creating some extra bias that you don't have with the first method. The last way, and that's by far my favorite, it's to do what we call fresh frozen. And I think it's because this is giving the more close to reality transcriptome and cell representation of any given tissue you're gonna assess. So in that case, you're gonna take the tumor out of the surgery room and immediately snap freeze it in liquid nitrogen. So you preserve the transcriptome of the tissue as it is at the moment of the resection. You don't have to wait 3 or 4 hours before doing that. And then once the tissue has been snap frozen, you can keep it like 4 years even in liquid nitrogen. And then at a given time later on, you can start doing the nuclei preparation with mechanic dissociation and perform your single cell experiment. The caveat of this is that you cannot do any more single cell enrichment because you just get the nuclei, so you cannot enrich for cell surface marker. You cannot do surface marker, surface protein interrogation with site-seq anymore, and you only get the RNA from the nucleus. But this gives you a way easier logistic and a non-biased dissociation of the tissue. One advantage of this also is that you can collect a cohort of many samples, which is really useful in the case of H10 for us. And for example, if you want to study, for example, a clinical sample from patients that have been relapsed to a cancer, you can collect the primary tumor and wait for the relapse and then only process sample of the primary and the relapse at the same time, which also limits some batch effect and confounding factor of preparing one after the other with sometimes months apart. So this is like a summary slide of sample preparation that you can do with the different methods I just present you on what are the things that can be achieved with each of them. When you do fresh suspension, you can basically do all of it. You can do single cell, single nuclei, of course, single cell attack. You can do RNA plus site-seq, which is your fast protein interrogation, and you can do also multiome from 10x. You can do the same thing with fresh frozen and fresh frozen suspension, and you can do fresh frozen tissue as well and do all these things except single cell. So when you do fresh frozen tissue, because you get the nuclei, the only thing that prevents you getting the nuclei to do it's working with the cell in single cell RNA-seq or single cell RNA-seq plus site-seq. So for the last like 10 or 15 minutes, I would just like to dive a bit more deeper in what we have been trying to do at Cell over the past years and what the team have been working on the wet lab side, and that has really helped us achieve higher throughput or more reproducible generation of data set. The first thing is we have been struggling for kind of some time to make nuclei preparation reproducible because everything was done manually before. And also it was kind of really time consuming and challenging to do this manually because every tissue we get, we have to almost spend an hour just getting to nuclei. But there is this company, S2 Genomics, that has developed this singulator, which is kind of a mechanical way to dissociate a tissue, snap frozen tissue, and generate nice single cell suspension. So now we are using that on a regular basis. We have even 2 of those in the lab. And that's how we get our nuclei preparation done. And we had really good results using this. And this has really helped us increase the throughput of the clinical sample we're handling now at Cell. And we get like really good nuclei preparation that are really, really reliable. And I'm just showing at the bottom, like the kind of optimization we have been doing with this kind of singulator to, for example, good, like better attack profile from snap frozen tissue. The second things we have been using is LeviCell, because one of the difficulties in handling clinical sample is access to fax sorting. A lot of those samples require to be fax sorted, and in our research center, getting access to an hour of fax has to be anticipated because it's heavily on demand. And now we have a LeviCell in the lab, which is a system that's allowing you to separate in a gentle manner a live cell. And we are using that. It's super easy to set up. It's fast. It can be done in 20 minutes. And one of the big advantages over fax for this assay is that it's way more gentle on cells than a fax. So we had a couple of samples like dendritic cell, or what I'm just going to present now, cells from the CSF, the cerebrospinal fluid, that are really sensitive, and when we fax them, they die. So using LeviCell is allowing us to handle those type of samples and making work more efficiently. So I'm just showing you like a quick video now of what's happening. So on the left part of your screen is a bright field. So basically you see the cell that have been loading in the channel. And on the right part of the screen, what you see is calcein staining, which is stain only live cells. The live cell with processed calcein and become GFP. So what you can see is like the live cell are gonna go at the top of the channel, while the debris and the dead cell are slowly going at the bottom of the channel. And then after 20 minutes, when we reach equilibrium, so of course the movie is like speed up here, we're gonna just flush and keep only the upper part, which is enriched for live cell, and we use this to process single cell assay. So you see, the more we wait, the more live cells are crowding at the top, and the more debris are going at the bottom. And now we just flush, and then we collect the upper part, which is mostly made of live cell. And the advantages of this is, like, as I say, the pressure inside the LeviCell is 100 times less than fax. And for really sensitive sample, we had found that this as being a game changer. So I just want to present this really little story in like 2 minutes, which is a collaboration we have with Adrian Boer and Jan Remzik, who is like a postdoc in Adrian lab, which is working on leptomeningeal metastasis. So this leptomeningeal metastasis is arising in about 5 to 10% of solid tumor. There is some specific tumor where it's like more frequently happening. And as of today, there is only few treatments that allow to treat for this metastasis. And there is only few labs in the world that is doing research on it. And one of the really specificity of those metastases is that those metastatic cells are surviving in a really harsh environment. So in a healthy individual, in the CSF, which is this like fluid in the surrounding the brain, there is almost no cell living in this, because the way this fluid is made really harsh for cell to survive there. And for example, there is almost no iron or almost no lipid there. But for some reason, in some cancer, some cell manage to metastase there and survive. And surprising thing is that also some immune cell will go there and be present when there is metastasis. So the main question of Adrian Boer lab is how do cancer cells manage to survive in this CSF fluid? And the tricky thing is that those cells survive in CSF, but when you take them out of the CSF, they tend to die really quickly. They are like almost adapted too much to survive in this kind of environments. But then when you get them out of it, they are not fit anymore and they tend to die. So that's kind of samples that are really tricky to work with because they can die quickly once we start to process them and isolate them from the patient. So also when we get them, the way we are doing this, the way Jan was doing it is like collect sample from mice, from model system from mice or from patient, and just isolate the cell and go to FACS. But he has to do everything really quickly, otherwise the cell would die. And also doing FACS sorting on those cells, for some reason, maybe the pressure inside the FACS would make them die. So we would sort LeviCell, but like 10 minutes after collecting the cell from the FACS, all the cells were dead. So there was no way for us to really obtain good single-cell RNA-seq from those type of cell. But when we try the LeviCell, we are like, okay, let's try this because first it's going to be faster instead of spending maybe an hour at the FACS to try to get those cells sorted. We can now do it in 20 minutes, and we can also do it in a way more gentle way because the pressure inside the LeviCell is way lower. And for us, using LeviCell in this type of really specific sample has made it possible. And this is, for example, one example of the data set that has been generating using LeviCell from, I think this is from a model system from mice, model organism from mice. And now we get really nice LeviCell and we get like nice data sets because LeviCell has been able to sort those cells. So I would say every project is different and every project requires probably specific optimization. And you really have to try to tailor the single cell method to the project and to your model system. I would also like to briefly mention, because it's almost time, these things we are testing right now, which is tissue grinder. We don't have a lot of data yet, but just for you to be aware, this is a non-enzymatic way to try to do tissue dissociation. So it's faster. It's done at room temperature instead of to be done at 37 degrees. And we have tried it, and for a couple of samples, we found that it's giving better results than doing enzymatic dissociation. It's not working for everything. We tried, for example, on zebrafish, and we find zebrafish cells are way too sensitive for this kind of dissociation, and they tend to be not of really good quality after dissociation. But we've done it on glioma, and in glioma, we find that we get nice single-cell suspension out of tissue grinder, and we are now in the process of trying to compare this more in detail to the classical enzymatic dissociation. So there is more and more way to process tissue, to process like a sample, and I feel that it's an exciting time to do single cell genomics. I feel I'm almost running over time, so I can try to speed up on the last few slides I have. This is something I really want you to keep in mind when you do single cell genomics, because that's the kind of question I can have is how much of the cell I'm going to bring to you going to get captured. So 10x Genomics is one of the best capture rate on the market, with about 2/3 of the cell you're going to bring to the core or to load on the 10x controller going to be captured, which is even better than if you do a plate-based method, for example. Another question I frequently have is how much cell I should target. And I would say there is no right answer. It's mostly depend of how big the population of interest is. If you are trying to study something that represents only 1% of your total cell population, so then you can need probably to target like at least 10,000 cells, because 1% of 10,000 is going to be just 100 cells. So 100 cells is kind of the minimum number of cells you need to do any statistically meaningful analysis. If the number of cells you want to target and study at the end is 50%, maybe targeting like few thousand is enough, because 50% of few thousand is going to be enough for you to do any analysis on those. So it mostly depends on what cell type you are interested in. The number of samples also is a question that is brought frequently to me. I would say it depends. Again, clinical sample tends to have a lot of variability from one patient to the other. So always aim for more than you would think, because you know that some will be bad quality and you will have probably to drop them. Some will be with batch effects, so you will have to correct for batch effects. So having more sample is always a good idea. If you work with model organism, you can do multiplexing. I did not touch base on that, but there is a way to pool several samples on the same experiment. And that's a nice way to increase number of samples you process without increasing the cost too much. Some also things that you have to keep in mind when doing sample preparation is have a good cell viability. Like some people don't mind running sample with 50% dead cell. It's not a good idea, because having so many dead cell in your sample won't give you good data. So even if you manage to select viable cell at the end bioinformatically, the data you get are not going to be good for one reason, that the dead cell release a lot of RNA in the buffer, and then when you encapsulate those live cells, you also have a lot of RNA from the dead cell, and this is going to give you a lot of background. You need a high cell concentration. You try to not get too much cell doublet or clumps, and also the buffer is really important. Don't, for example, use FBS, because FBS is full of RNAs, and this is going to have an impact on your RNA quality. So I'm just gonna skip through this quickly, because I don't think I have much time. And just to touch base on the things we have also tried to develop beyond just doing a single cell RNA-seq from 10x is try, for example, to rescue signal. So we call this target enrichment, and this, for example, is an example of a gene from a collaborator that is lowly expressed. So you can see in the original data set, the gene is barely detected. You can bioinformatically do some expression inference and try to recover expression from those, from some of those cells by looking at neighboring cell and see which one is expressing. But we are also doing these things, which is now what we call target enrichment, which is trying to rescue expression for some of those cells, for this gene in some of those cells. And you can see that on the right panel, you have a nice correlation between bioinformatic inference of this gene expression and then the a wet lab experiment that's trying to rescue this gene expression. So there is things you can do if you have like difficulties capturing the signal you would like to capture with your single cell genomic experiment. So don't hesitate to explore. There is new method and new protocol almost every day now getting published. So there might be something that might help you answer your biological question. And I think I'm going to leave it to that. I would like just to finish by one last quote. It's not from me, but that's one I really like. And the work we do at Cell every day, that I would rather be an helpful academic than an important one. And that's who we are. So even though you are not at MSK and you have some question and you want to reach out, for any reason, don't hesitate. We are always happy to help and try to answer the question or the kind of difficulties you are encountering in your work. Of course, I want to acknowledge everybody we are collaborating with. I am director of Cell, but Dana Perry is the scientific director of Cell, so I want really to highlight this. She's like really an important person helping making Cell a nice facility for everybody at MSK. I want to thank all the collaborators, and most likely we have open position in the wet and dry lab. As I showed at the beginning, single cell is really expanding, and we have more and more demand. So we have open position. Please don't hesitate to reach out to me if you're interested to join the team and do some of those work with us. We'll be glad to interview you. And just to finish, I really want to put a picture of the team, because I'm talking today, but they are the one doing the work every day, and they're all amazing at what they are doing. So I really want to finish showing them once again. And thank you, everyone, for your attention and for sticking around with me till the end.
Great. Thank you, Ronan. As a reminder to our webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask all attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. I'll now get into some of the questions that have come through already. Given the challenges with tissue dissociation, how meaningful do you think 10x's fixed RNA profiling kit launch will be for overall single-cell market growth and use in your lab and others? Is that a product you're familiar with?
So I guess what's mentioning is a PFA transcriptome kit that's going to be released by 10x. So there is different things you have to consider. Is like, is the fixation going to be done before or after tissue dissociation? And I'm not sure that 10x is going to come up with like a method to do dissociation on tissues that have been paraformaldehyde fixed. But one thing I know is that this kit's going to be not relying on polydT capture for assessing transcriptome profile of the cell, but going to rely on probe targeting gene specifically. So because when you fix with paraformaldehyde, you're going to chop up your RNA in small pieces. If you would do polydT capture for those RNA, you tend to not get great signal because most of the RNA will be fragmented. So the way to work around this for 10x and for other methods when you have a tissue that has been paraformaldehyde fixed, it's to come with probes, short probes that will bind at different location on your RNA, and then even though it's chopped in pieces, you still get signal for the RNA. The things, and we're gonna probably like compare and benchmark in the months to come, is like, is the expression dynamic you're gonna get from the PFA kit from 10x gonna be really comparable to the polydT capture? We know that, for example, even though it's like full transcriptome capture, if you do the 5' or the 3' kit, it's not always to integrate the data set from both kits because the capture efficiency is not the same, whether you capture by the 3' or the 5' end of the gene. So my feeling is that if now you do 3' polydT capture versus probe capture with a PFA kit, there might be some difference in the gene expression dynamic. So you just have to be aware of that, but definitely like fixing the tissue early on would probably prevent a bit of like the bias we see when we do single cell dissociation enzymatically. But other caveats will erase at that moment.
We have another question. I think you wound up getting into this a little bit after the question came in, but what is your mechanical dissociation approach?
So the mechanical dissociation approach is like really easy, and that's really basically smashing the tissue. So I'm not sure which mechanical dissociation the person is mentioning too, but I guess it's based on when we do nuclei. So the way it's done when you do nuclei dissociation is really snap freezing the tissue. So snap freezing the tissue in dry ice or in liquid nitrogen will like basically break the cellular membrane of all the cells. So it's already kind of achieving part of the job at mechanically dissociating it. And when you get your piece of tissue, whether you do it with the singulator or with like manual dissociation with like a dense homogenizer, basically you're gonna like mechanically disrupt cellular membrane. And because nuclei are like stronger and like resistant to this mechanical dissociation, you're gonna preserve the nuclei, but all the cellular membrane and all the cell gonna be disrupted, and that's how you release single nuclei from tissue. So it's and the advantage is also with mechanical dissociation is that it tend to work with any tissue without further optimization maybe in the buffer, but not in the mechanical dissociation itself. When you do enzymatic dissociation, you have to adapt the time, the temperature, the cocktail of enzyme to every single tissue. And even within a tissue, using different enzymatic cocktail will give rise to different cell types you collect. Like it can be more immune, more tumor cell, depending on the enzyme you use. When you do mechanical, you tend to just smash all the cell and you recover all the nuclei from all the different cell types, doesn't matter which cell type is. So that's really why it's not as biased as doing enzymatic. And that's also easier because it requires less optimization at the end of the day than doing enzymatic dissociation.
Have you considered using tissue storage solutions prior to cell dissociation?
We never try that. because I guess it's just gonna make it easy on the logistics somehow. So doing tissue preservation buffer will just like prevent you from having to do the dissociation just after surgery. So yes, it's kind of make it easy on you for the logistic, but at the end, it won't remove the fact that you're creating some bias when you do the tissue dissociation later on. So we never really tried because we feel that it was not really a way for us to move forward and to prevent this bias we have when we do single cell dissociation.
This viewer asks, have you tried cryoprep or pulverizer methods on flash frozen tissue?
No, we don't. Never tried that.
The next question here, regarding the cell buffer composition, what should you think about when it comes to FBS?
So FBS is like, as I say, it's really full of RNAs. I know it's good for the cell and it's really can help them feel more healthy, I would say. But that's something like all the protocols are really preventing people to use because FBS is always RNAs full. And when you do single cell RNA-seq, it's never a good idea to have a buffer with RNAs. So we really want to have people that we work with to not use FBS and instead use PBS plus BSA, or you can even use like DMEM or like kind of commercial media with like control composition, which would make the cell feel more healthy and survive longer till we do the encapsulation. But yeah, FBS is definitely not a no-go for us.
We have a couple of questions, a couple of similar questions here. We're producing nuclei manually and have concerns about clumping. Do you have any suggestions?
So I would say we have some tissues that generate more clumping than others. And something we have observed basically with manual dissociation is, so I tend to say that nuclei are more resistant to mechanical dissociation, but they are not also like resisting to anything. So something we have observed when we used to do manual dissociation is that we had variability from one operator from another one between operators. If one would be like, let's say, more diligent at trying to smash the tissue, we would have like maybe more like debris formation because the down homogenizing was done stronger than another person. So I think for clumping, one of the issue might be that if you smash too much nuclei, you're gonna have like some DNA release, and the DNA will tend to make pellet and then favor clumping. And that's why switching to S2 Genomics singulator has been really game changer for us because we kind of not get that anymore, only on a rare occasion. And to come back to clumping, it also depends on the tissue. Some tissue, for example, like brain tissue, are full of myelin and kind of favor clumping. So there is kind of specific things to consider depending on which tissue you are working with. And I would say sometimes more is less. So if you want to do nuclei, don't be afraid to start with really tiny piece of tissue. Like tissue can be really high in cellular content, and you don't need big piece of tissue. Using a big piece of tissue might have like a kind of negative effect on your yield and favor like bad quality data because you will have too much debris. And the yield will be lower. So maybe starting with something like it's 10, 20 milligram only in size, using like bigger buffer volume might help with clumping. But I mean, if the person have like difficulties, they can reach out to us and we might be able to give you some tips on help, depending on the tissues they use.
Great. Unfortunately, we have a couple more questions here, but that is all the time that we have. We'd like to thank Ronan Chaligne and our sponsor, Levitas Bio. If we didn't get time to get to your question, we'll try to follow up. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of the webinar, if you'd like to listen to it again, an archived version will be emailed to all attendees. And thank you so much for joining us.
Thank you for being here.